Hello, in this presentation I'll introduce a comprehensive method for assessing aquatic ecosystem health using mayflies as biological indicators. I'll cover why they were chosen, how sampling is conducted, and stresses like sewage and agricultural runoff are influencing this ecosystem. Rutland Water is one of the UK's largest reservoirs. Created in 1975, it is used to supply water as a reservoir and acts as a recreation hub, a nature reserve, and a site of interest. However, intensive farming and nearby urban areas contribute treated and untreated sewage, as nutrients, sediments, and pollutants that threaten water quality and affect the quality of the habitat. This map of which represent water flow. Green arrows represent clean water from the reservoir. Yellow represents treatments and red signals farmland runoff. Lagoons 5 and 7 are most likely to be more polluted because they are closest to farmlands. So why should we monitor aquatic health? Well, monitoring allows researchers to track ecosystem progress and highlights key changes so that management techniques can be improved. Pollution causes an excess of nutrients like phosphorus and nitrogen, which creates perfect conditions for algae to bloom, which eventually leads to eutrophication, which is where great concentrations of algae and microbes develop on the surface of a water body, decreasing light and oxygen. Regular assessment and consistent management will prevent this from happening. Biological monitoring using invertebrates as indicators is a very cost-effective and practical way to measure water quality. Methods such as BMWP and ASPT may help to prevent these problems. Mayflies belong to the order Ephemeroptera and have over 460 genera. They are systems and prefer shallow depths, making Rutland a suitable habitat. They act as primary consumers, detrimental food source for fish and larger invertebrates. They have been chosen as they are typically present in Rutland area naturally and aquatic environments. They contribute to up to 25% of total zoobenthos production. Mayflies that occupy various niches, with some species grazing on algae, others filter organic matter, and some even predate. They contribute to multiple food webs, sustaining the ecosystem and occupying various functional roles. Sensitive to pollution. Sensitivity means pollution can be detected early, although they cannot infer the severity of pollution for improvement before devastation of the ecosystem. They spend most of their life cycle submerged, meaning they deplete habitats. Mayflies undergo various life stages in aquatic environments and so are differently affected and sediments at each stage of their life. They undergo incomplete metamorphosis, consisting of egg stages. Mayflies occur at subsurface zone during the early stages of their development cycle and then the final themselves from predators or harsh conditions. Adult stages occur up to 10 centimeters deep in sedimentation. Many mayflies are positively thigmotatic and spend most of their life cycle in a hypogeal environment. What makes mayflies so sensitive? Larvae usually feed on detritus, so are likely to be exposed to substrate or an increase of toxins in the water. Mayflies produce lots of eggs, but the rate of egg growth is affected factors, so they can indicate environmental conditions. Most mayfly larvae have specific requirements to oxygen, current, and temperature. These requirements explain their decline in cases of eutrophication, sedimentation, chemical pollution, climate change, and overall water quality. Heavy metals, pesticides, pharmaceuticals, micronutrients all contribute as pollutants and affect mayfly growth rates, mortality, reproduction, and affecting both mayfly populations and other trophic levels in the Rutland food web. They are particularly vulnerable to climate change, which results in smaller adults and lower reproductive rates. 67% of mayflies have. This means they cannot migrate to better temperate conditions like larger invertebrates can. This makes their numbers particularly. Notably, Amelitus inopinatus is a particularly threatened species, with its ecological decreasing as temperatures rise, isolating populations and preventing genetic adaptability to these stresses. So what is BMWP and ASPT? BMWP is the Biological Monitoring Party score. It sums pollution sensitivity score of all macroinvertebrate families found in a sample. High scores indicate better water quality, however, it can be biased by sample size and taxa richness. ASPT is the average score per taxon. It is the BMWP score divided by the number of scoring taxa, providing standardized measure of pollution sensitivity. It allows for better comparison between sites by reducing sample size bias. Mayfly's short life cycles make them great indicators of water quality. They have a BMWP score of 7 to 10, according to different sources. The method chosen is catch and release of mayflies by kick sampling. This will take place across lagoons 2, 3, 5, 7, and 8, which are in different areas of Rutland Water, providing a representative sample. Kick sampling involves placing a 1 mm mesh D-frame net in a shallow vegetated habitat, as well as other microhabitats. The net should be held against the current to ensure swift flow of insects into the net. The substrate is kicked repeatedly for 3 minutes across multiple microhabitats, such as shallow and deep areas, high vegetation areas, rocky or muddy areas. Sampling will take place every 2 weeks for 12 months, starting in May. This is to ensure mayflies can be present as they typically emerge at this time. Invertebrates are then placed into a white tray and identified. This can be done on site or in a lab. Specimens will be released unharmed. Smaller individuals may need to be transferred into a smaller disc for identification. This technique is minimally invasive and ethical as individuals are released unharmed. Samplers will take care to ensure the safety of specimens. Identification is helped by the use of field guides and mayfly keys to family and genus level. Specimens will be identified to species level where possible to provide a broader data set and accounts for more pollution tolerant species. Mayflies are typically distinguished by their gill plate shapes, having 3-point tails and their leg segmentations. The field data sheets will record mayfly presence, abundance, their BMWP and ASPT scores. Water temperature, pH, and chemical levels will also be recorded by using devices such as digital thermometers, digital pH meters, and colorimeters. Mayflies will be quantified according to density per litre of water. Size of individuals will be measured as well as population structure, recording the presence of adults, larvae, or eggs. This data will be compared across sites. Statistical software will be able to correlate mayfly densities with nutrient levels, pH, temperature, and site BMWP and ASPT scores, and chemical concentrations. At Rutland Water, there are various anthropogenic stresses like agricultural runoff from fertilizer and pesticide use and sewage from nearby urban areas. Recently, in February 2025, a residential oil spill contaminated the reserve. Farmland runoff introduces nutrients like phosphates and nitrogen, harmful chemicals, and sediment. The nutrient excess leads to algal blooms and eutrophication, depriving oxygen in the habitat. Even treated clean sewage reduces oxygen levels and releases hormones, chemicals, and pharmaceuticals. Mayflies are often one of the first taxons to succumb in these conditions, signaling an ecological stress before algal blooms occur. The loss of mayflies therefore infers that there is a problem within the habitat. Recording their abundance therefore helps to address where management needs to improve before it is too late. Mayflies have a BMWP score of 7 to 10, making them crucial indicators of aquatic system health. Water. This method is practical and applicable globally. It helps to detect pollution early, preventing further irreversible damage. It tracks the success of existing strategies and highlights areas that need improvement. It also provides a scientific basis for guidance on policy changes and decisions. However, BMWP is commonly used, but it is a limitation that it was developed primarily for flowing water systems like rivers or streams and has reduced accuracy in lentic environments. It only considers the presence of taxa, not the abundance, but in this study this has been mitigated by counting abundance. It's also mitigated by the combined use of ASPT, which gives a measure of tolerance that isn't influenced by how many taxa were counted. A study by Armitage in 1983 found that ASPT is less influenced by season than BMWP is. indicating samples collected in any season will be consistent if using ASPT scoring. It is also less dependent on sample size. The use of ASPT significantly reduces the limitations of BMWP alone and significantly strengthens the method. It also allows for site comparisons and better applications globally. Broader diversity when sampling can be improved by the use of EPT index or identification of moderately tolerant species in combination with mayflies to provide a balanced assessment. Deployment of automated sensors to monitor temperature, pH, turbidity, and oxygen levels to track short-term pollution events may detect triggers early. This study will try to identify individuals to species level, but BMWP only provides data for broad taxonomic groups. Mayflies are all grouped together despite some genera being more pollution tolerant. Additionally, these methods don't account for other stresses such as microplastics, chemicals, and thermal pollution. Rutland suffers from this type of pollution, which is why this is mitigated by taking data for these stresses. Although mayflies are good indicators to detect pollution, their high sensitivity limits their presence in moderately polluted areas, which doesn't allow us to assess how degraded a habitat is, only that it has some pollution. This is perhaps a good thing, as it stops pollution before it gets worse. But this creates a zero data problem, as with the indicator missing entirely, we cannot determine how badly an area is polluted, and so efforts cannot be prioritized. Mayflies can't infer the degree of degradation, so additional taxa such as caddisflies may mitigate this. This infers that BMWP and ASPT scoring should be accompanied with other methods like the EPT index. EPT assesses stoneflies and mayflies, which are highly sensitive, as well as caddisflies, which are more tolerant species, providing a better picture of the degree of degradation. Using mayflies at Rutland as indicator taxa is appropriate as they naturally occur in the Midlands, and Rutland water has various microhabitats within the lagoons, making them an ideal place for mayfly development, which can support the presence of pollution if they disappear. Their high sensitivity indicates changes. The method of kick sampling and catch and release is able to be completed with a small group of volunteers and is non-invasive. It does not harm the mayflies and is cost effective. Long-term changes can be understood due to the long duration of the study and will identify key areas that are most effective and patterns. The results could even identify a cause and will emphasize a need for improved strategies and understanding of how unexpected events may influence data. BMWP relies on species richness, but this method also measures ASPT, meaning we can assess whether results are due to data deficiency or pollution. These factors, along with collection of physiochemical data of water and mayfly abundance and size, mitigates the limitations of using just BMWP scoring alone. Multiple lagoons will be surveyed, ensuring a broad coverage of Rutland so that high polluted areas can be identified. The data produced is easily repeatable and is simple to carry out. BMWP is easily interpreted and is widely used, so the method can be applied elsewhere. It already exists in national water monitoring frameworks. This method is adaptable for future studies and allows for inclusion of additional taxa like EPT and helps to create new conservation priorities. My hypothesis is that lagoons 5 and 7 will not have mayflies present due to runoff from drainage pipes and nearby farmland. High concentrations of chemicals and nutrients create conditions where mayflies cannot develop. Mayflies can't develop in moderately polluted habitats. This will likely put pressure on authorities and conservation agencies to create new strategies and policies, guiding nearby farms and landowners to follow new ethical techniques and reduce the use of fertilizers and pesticides. The results will provide data-centered guidance and provides a standardized surveying technique that can be applied to other taxa and locations. The loss of mayflies will likely affect other trophic levels and reduce overall ecosystem health. It is important to use mayflies as early pollution detection is easier to manage and control. In the other lagoons, presence of mayflies is predicted, but in higher polluted areas, low numbers of small adults are likely to be observed. This is because pollution and climate change affects mayfly growth and development, resulting in less successful smaller adults. Applications from these results will result in early pollution detection, will prevent further degradation, tracking success of existing management techniques, and provides a scientific basis for policy changes and management decisions. Stakeholder engagement. Policymakers and environmental regulators, such as Environment Agency or Nature England, are responsible for enforcing regulations and managing nutrient runoff. They can incorporate mayfly monitoring to detect early pollution and contaminants. They can use the long-term data to justify implementing new policies to reduce pollution. Conservation organisms can integrate mayfly monitoring along with other taxa and combine the techniques into broader aquatic assessments. They are able to use the data to apply for grants and produce evidence based priorities. Scientific communities and educational institutions use Rutland water as it is an ideal environment to study pollution, its effects, and the impacts of other stresses like climate change. They can incorporate results into undergraduate research, school field trips, and community STEM initiatives. This increases overall engagement, increases data, and allows for institutional collaboration. Benefits from this study include data that will inform restoration projects for conservation organisms. Mayfly abundance typically indicates successful wetland management, so data supports project proposals. The data will overall contribute to cleaner water for the reserve and for recreational activities, boosting funding and public engagement. Loss signals early warning signals of pollution, helping to guide operational decisions for water management authorities. Biological monitoring data can be integrated to existing sustainability programs. These authorities are responsible for changes. This data helps set the standard of when intervention is needed. Data identifies pollution rich areas and will help create sustainable farming techniques. The identification of where pollution is highest puts pressure on farmers to reduce fertilizer usage and pesticide usage, encourages buffer zone development and filtration areas such as areas with high vegetation. Future directions from this study include encouraging community participation to increase sampling coverage, increasing surveying of invertebrates to reduce pollution early and manage any changes, increase education, which will spark engagement and increase funding, and it influences buffer zone development and improve policies, providing evidence based guidance for farmers and landowners nearby. Thank you for listening.